This video on our course B01 Data Management, we'll talk about the key concepts around that next. So we consider then in terms of what's research data. So that will be the source data, all data that's collected, created, and used by research, including data else held elsewhere. Assembled data sets are included here, so that can be data extracted or derived from the above. Reference data as well, so any subset of the above used in your analysis or to draw conclusions, including supplementary material. And this, for example, can be observational data, experimental data, model simulation data, derived integrated data sets, and curated data sets as well. So we can think about then what is the structure of our data as well. So we may have, for example, a particular sample that's associated with a particular metric, and other data sets that are linked to this as well. We think, how do we normally structure that database? So any data is any information that's collected, stored, and analyzed to give insights to inform decision making. There's various forms, including structured data that we're most familiar with. That's a structured table. So like a list of names, ages, and birth dates, for example. We may be also familiar with semi-structured data that has a varied structure, that it may have systematic identifiers inside that, but it may be more flexible to accommodate different formats. For example, if you downloaded a GenBank file from NCBI, that's a semi-structured data set. And then we have unstructured data, and that may be not in a specific organized manner. Images are a good example of that. So these data sets differ because structured data is a predefined format that is intuitive. It will be stored in a relational database, and the analysis is relatively straightforward, but it may not necessarily be scalable to different areas. Unstructured data set may have a partial structure, but it may not be clearly predefined. These are typically stored instead in a file system or a data lake. Analyzing this can be complex, and we use particular tools. So, for example, analyzing a genome sequence file, a genome read library, or a series of images would be examples of this. And then we also have semi-structured data that has some structure but varies a lot. And that these might be stored in file systems or particular databases, and requires some interpretation because if you're trying to understand multiple files in this format, it's not necessarily straightforward. So you're familiar, I'm sure, with structured data already. So these are structured into rows and columns. Typically, the columns contain information of the features of the data set, and the rows might be items within there. It could be samples, for example. And what's most important is that each column contains data of the same type. So either numbers or dates or text or whatever it is. We're not mixing those within the same column. The structure should be predefined and consistent and follows existing nomenclature. If we look at semi-structured data, that's more varied structure, may contain tags of markers that are not present in unstructured data, and those could act as delimiters to separate it out. So the arrangement of the data is not fixed. It can vary in being very short or long. There may be known markers that help you define the start and end of different things, and may be quite nested depending on the complexity of the data set. Semi-structured data may be studied in different file formats. JSON files will be one example, and XML files will be another example, and that those require particular approaches for interpreting that data. So you think of structured data as having a clear, predefined shape, where semi-structured is a intermediate, where you might have examples. So this is an example of a JSON file containing a sample, and then you see here we have information on the chromosome, the position, the retinal alternate alleles, and the type of mutation that's present. But you see then the next entry has some of that information, but not all of it. So you can see that they differ slightly, and it is, if you're reading this, it is clear what the information contains, but we know that we don't necessarily have all the information all the same information present for every particular instance in this data set. So often these might contain, for example, mutation files from omics-related data sets. Though they may be stored in FASTA or FASTQ formats. XML can also be used as well. If you're looking at, say, biological pathways or networks, they might be also stored in XML or JSON. And the same applies for gene expression data as well. If you've ever looked at GenBank files, that's an example of semi-structured database, or you have an accession ID at the top here. You've got length. I know things like what type of molecule is it, a DNA here. And all the information on the accession IDs, the versions, the sources, comments, and other things. And if you look at different GenBank files, the arrangement of this is in the same way, but not all entries here may not necessarily be present, and they may have very varied levels of detail. And then if you move on to unstructured data sets, if we look at the example in sequencing or bioinformatics, it might be raw sequence reads, logs from experiments, or other data sets that may not have any predefined format at all. So we're looking at things something with no fixed format, does not follow a particular organizational structure, such as rows and columns, is very diverse data types. So it might be video, audio, text, emails, and it is complex to process. So this might require advanced techniques. So examples of this would include text documents. So for example, a Word document would actually fit into that category. Images are other examples of this. And this means that having a searchable database is not necessarily straightforward. And then managing this data might require a specialized system or a specialized database. So an example of this are FASTQ files that are technically semi-structured, but the formatting within there is very varied. So you typically a first identifier which may contain more information that's not shown here. So this line might be quite long. Then there's a line containing the sequenced bases. We have a plus on the third line, and then on the fourth line you have the quality scores referring to the bases above. So you can see here that when we go to other lines that have the same overall structure, but the information level that's present will vary a lot. So you see this other examples are images such as microscopy files, gel images, and so on. So that concludes this part on key concepts in data management.